So good morning, everyone, and thank you for joining us today. This is the third installment in the seed training program by Tyfu Cymru. I'm sure you're aware today's topic is selection of crops for seeds, and we're welcoming back Katrina from Garden Organic Heritage Seed Library. If you were unable to join the previous two webinars, just like this one, they were recorded and they are available on the Tyfu Cymru website on our Knowledge Hub, so you can take a look at them there in your own time, and this one will be uploaded on there after the webinar. Just give us a few days to get that up there. As I'm sure most of you are aware of the work that Tyfu Cymru do, but if we've got any new people here today and new people to the project, Tyfu Cymru delivers business support and training to commercial horticulture enterprises in Wales. So if you fit that description, then we are able to deliver fully funded, 100% fully funded training for you and your business. And one of the benefits of Tyfu Cymru is we can tailor it to exactly what your business needs are. So if there's something specific that you would like support in, then please get in touch with us. We always love to hear from you, and we can discuss how best we can support you going forward. So you may have seen this before if you've been in our previous two webinars. There's a few housekeeping rules which I'm just going to whiz through. So all attendees, you're muted with the camera off, so we can't see or hear you. So if there are any issues, then please use the chat box to get hold of us. Today, we're actually going to have the question and answer section at the very end. So if you think of any questions as you go through, then please leave them in the question answer box below, and we will get to them at the end for Katrina to answer. We will be recording this session and we will be posting it on the Knowledge Hub, and you can find that on our website. And if you do accidentally leave the webinar, if you have any tech issues or what have you, you can rejoin us using the same link that you joined first thing this morning. So I think on that note, I'm going to hand over to Katrina to go through today's webinar.
Welcome to this third and final webinar on crop selection for seed production. If you joined us on the first two webinars, you know that we looked in some detail at some of the crop cultivation requirements for open pollinated seed. This particular webinar is going to look more closely at the process and principles of selection in order to produce high quality open pollinated vegetable seed. So back to those two simple rules that we've been exploring so far. In order to produce quality seed that's true to type, we need to do two things. Firstly, we need to prevent cross-pollination, and secondly, we need to save seed from only the very best examples by removing any undesirable individuals along the way. And it's that process that we're looking at in terms of crop selection at this webinar. So we'll be covering these areas: understanding population size and its importance to inbreeders and outbreeders and what that means, what is true to type, and how do we know what we're doing when we're selecting, what are we selecting against, and we'll be looking specifically at the role of selection and roguing for inbreeders, outbreeders, and specifically what's involved with biennial seed production. And we'll also be looking at the way we look at seeds beyond the harvest stage, so that we know that we're saving only the best examples of those seeds. And remember, we're dealing here with open pollinated varieties, so essentially ones that will breed true to type with a bit of care from one generation to the next. We know that pollination can occur by vectors such as insects, wind, or by hand, and the process of isolation and pollination is something that we've already explored. The seeds themselves carry the parent's genetic material, but what's important about open pollinated varieties is that they are inherently variable, and that those variations can be quite slight, for example peas, or they can be quite noticeable, such as brassicas. And it's that managing that variability which we are looking at from a seed selection point of view. So growing for seed saving requires those methods that are going to maintain those distinctive features of that variety by controlling the seed production within that variety and using the process of seed selection in order to do that. So let's look firstly at the role and importance of population size in this process of growing for seed. Inbreeding is itself quite useful in terms of retaining desirable characteristics or eliminating undesirable characteristics, but a population size that's too small often results in decreased vigour, fruit size, plant size, fertility of the offspring, etc., because of the combined effects of genes that have been recessive in both parents. So when we talk about inbreeding depression, actually we're talking about reduced biological fitness. And that refers to the plant, the organism's ability to survive and perpetuate its genetic material. So inbreeding depression is often the result of some kind of population bottleneck. And as a general rule, the higher the genetic variation or the available gene pool within a breeding population, the less likely it is to suffer from inbreeding depression. So depending on the vegetable type, that population number is going to be very different, but it's essential, that population size is essential to maintain a healthy and genetically diverse population. So for inbreeders, and we'll talk a little bit about that, self-pollinators, this is less of a problem. However, for natural outbreeders, continuous inbreeding due to a small population size is going to result in inbreeding depression. So in terms of planning, well, you need to consider population size from the very beginning, because the number of seeds that you choose to sow from the start must take into account the number of plants you're going to lose along the way and the final number of plants from which you're going to save those seeds. So you need to consider these particular points. Firstly, know whether they're an inbreeder or an outbreeder. I'll be looking at some specific examples, but I'll also be directing you at the end of the webinar to our online resources. So if you're not sure and we're covering a vegetable type that's different to the ones that you're growing, you can look up the references and they'll give you some idea of whether they're inbreeder or outbreeder and the population sizes you need. You need to consider the seed that you currently have, the age and the quality of it. You need to accommodate losses due to pests and diseases along the way, and we'll look at why that's particularly important for biennial seed production and the planning involved for population sizes there. And of course, because they're open pollinated and you're only selecting from the very best examples, you need to accommodate how many examples you're going to be selecting out from this process. With all these principles in mind, and having accounted for population size, and just to give you some idea of range, for inbreeders, which we're looking at more closely in a moment, we might be only talking 1 or 2 plants for a healthy gene pool, whereas something like these radishes on the right hand side, which are highly variable outbreeders, we could be talking of a population size of at least 100 plants to save seed from. So once you've accounted for those, we are going to be looking at how we decide what are the best examples to save seed from. And that process continues throughout the growing phase, right the way through to the seed itself. So it's a given that you're going to be removing any unhealthy looking or diseased plants along the way, and obviously you'll need to do your own research if you come across diseased plants to ensure that you're not dealing with something that is seed-borne or some other issues that are going to cause problems with your quality crop in the process. However, we do also want to maintain the specific characteristics of the variety that you're saving the seeds for. So how do we know that what we're looking at is correct for this variety? How do you know what you're going to select against as you're growing it? And this is really important because if you're going to be producing quality consistent seed of a particular variety type, then you'll only want to be saving from the best examples, and you need to know what that best example looks like. So a little bit of background in terms of what we do at the Heritage Seed Library. We're maintaining a collection of around 800 vegetable varieties, so we really do need to know what we're growing, and each year we grow out somewhere between 100 and 120 varieties on site, but they usually include around 10 to 20 trial varieties, and these are either our varieties that we are exploring a little further, or we need to investigate because we have some question marks over them, or as in the case of this slide here, these are going to be varieties that we haven't seen before. They are potential new additions to the collection, potential rediscovered heritage varieties, and we go through a very specific process of characterisation to produce a description sheet for this particular variety. So I've got this one up here, Skipper's Mummy Pea. We're growing it out this year. It's pretty typical of some of the tall tales and lovely stories we have behind our collection. So this heritage variety was named after Miss Skipper, who apparently was a missionary in Africa many years ago, and the claim to fame, the rather tall tale, is that she was given these seeds from a pea that apparently was discovered in an Egyptian tomb at some point and had been grown by the family in Norfolk ever since. Clearly not quite how it would happen, but as a possibly dating back to the 1920s example of interest in Tutankhamun and Egyptian tombs at the time, this is a really interesting heritage variety for us to investigate a little bit further. So you can see that when we grow these varieties out, we are making a full assessment of what they look like, and in their first year, their trial grow out with us, we collect all sorts of information about the varieties. The sheet on the left is something that we put together for each of our vegetable types. There's usually several pages worth of information, and the characterization categories themselves are based on a combination of bioversity and UPOV descriptors. So UPOV is the International Union for the Protection of New Varieties of Plants, and they have produced sort of universally recognised characterisation sheets. For us, that's important because we're dealing in non-commercial varieties. We're dealing in what might be slightly genetically wobbly heritage varieties. So for us to be able to make an assessment of them and decide whether they are similar to what might be already available on the market as commercial varieties is important to us. Obviously, once we've got this information, we can then make sure that when we grow it out again to regenerate the seed supplies, we've got some reference point to look at. Clearly, for the scale of varieties that we're trying to maintain, that's a particular process that we use that works for us. Obviously, for yourselves, you may want to take a different approach, but of course what you must bear in mind, if you are aiming to produce seed on a commercial scale for commercially recognised and commercially listed seed varieties, you do need to make sure that the seed that you're producing is as described for that variety. So we're looking specifically at inbreeders, and really for many crops that are simple to save seed from, and we've covered those in the first webinar, the French beans, the peas and the tomatoes of this world. In terms of selection, you wouldn't expect a huge amount of variation within a population. So for peas, for example, you could start off with 10 plants and easily afford to rogue down without the issue of inbreeding depression. Of course, if you do end up with just one plant that's true to type or healthy, you will want to understand the decisions at play to get to that level of reduction. And if there was considerable visible variation across a population of inbreeders, then you might suspect that something else is at play, whether it's cross-pollination or some other kind of seed contamination. Let's apply these principles and work through a scenario that's also going to build on the knowledge that we've built up from the first two webinars, and it's helpful for you to perhaps refer to the seed saving guidelines via our website, which I'll direct you to for some specifics on this. So we're growing this fictional lettuce, Right and Red Splash. We're going to grow it for seed, and we need to establish whether, first of all, it's an inbreeder or an outbreeder. So we know that lettuce is an inbreeder. It's will only require a very small final population of perhaps between 1 and 2 plants to make sure it has a biologically fit genetic pool. Lettuce, it has itself perfect flowers, which means that it can happily self-pollinate. There's a low risk generally of cross-pollination, but that is subject to a recommended isolation distance of 8 meters. So you've accounted for that. We know that the seed is pretty good quality, but it's 8 years old. And for lettuce seed, if kept in the right cool and dry conditions that we've previously mentioned, the germination rate will begin to reduce after about 7 years. So that's something we need to bear in mind. Lettuce itself can be susceptible to diseases such as botrytis in particular, so we need to allow for some of the losses along the way, and we decide to start off by sowing 10 seeds. 8 out of the 10 seeds germinate, which is not bad for seed that's 8 years old or more. We remove 2 plants along the way, so when we talk about roguing out, we are literally removing the plants through this process. So 2 botrytis plants have been rogued out. We also have some signs of bolting further along the way, so 2 plants have flowered earlier than anything else, and we don't want to be saving seed from bolting plants. The seed that produces will also include a tendency to bolt as well in future generations, so we take them out of the process straight away. They're down to 4 plants, and we rogue out 1 plant because it doesn't have the telltale red splashes on the leaves that we're expecting for this plant, so we know something about how it should look. We've got characterization information, and we know it's been described as a loose leaf lettuce with distinct red spots on its green leaves. So should we discover something that is not true to type, we are going to rogue it out and only select those best plants to save seed from. So when the plants are mature, we decide that although we're left with 3 plants, we know that the yield for lettuce seed is going to be pretty high, and we can afford to save seed from just 2 of the healthiest plants. We could save from 3, like I said, the larger the population the better, and because of our purposes for saving seed, and we know we've got the 2 best examples, that's where we end up producing seed from. And I think it's worth saying at this point that the scenario that we've just worked through very much reflects the working practices that we conduct at the Heritage Seed Library. Clearly we're a slightly unusual organisation and conservation charity rather than a commercial seed producer. So from your perspective, as someone who may be planning to or is already producing seed for commercial sale, you will want to be looking at the kinds of population sizes that reflect the yield that you need as well. So clearly we're operating and purely driven by the requirement to replenish the core library seed sample for many of our heritage varieties. From your perspective, you are likely to be specialising in a much lower number of varieties each year, but clearly you will want to be reflecting the quantities you need in the crop rotation plans and the growing spaces that you have for growing each year. So let's look at outbreeders. Clearly significantly more complex perhaps than the inbreeders, and we know from the second webinar that we were addressing some of the trickier crops in terms of isolation and pollination. We're talking here about things like onion, leek, carrot, brassicas, broad beans and runner beans, as well as squash, and wind pollinated crops such as sweet corn, beetroot and spinach. They're all crops that we considered at the last webinar. But selection for outbreeders follows the same underlying principles as inbreeders. So you are going to be using your knowledge about the crop, about the variety itself, perhaps using a descriptor or characterization photographs to make very specific selections and removing any plants or products with atypical traits. However, as we discussed, our breeders are going to need a wider gene pool to avoid inbreeding depression. They'll require higher population size than inbreeders, and there will be more variation across an outbreeding vegetable variety, and this does need to be recognised when making a selection. You may also want to be roguing out or removing traits that are universally undesirable for a vegetable type in general. So these could be traits that make it less appealing to eat or harder to store, for example. So we could be talking about varieties that bolt early, so you want to be selecting out from those. Or there may be a high proportion of leaf to root in root crops, for example, or flat or concave shoulder in onions, which means that they're more likely to rot in storage. Or it could just be that you want a less hairy radish root, because otherwise that would make it harder to clean and eat. So other considerations for vegetable types themselves that are worth looking at. So let's just briefly look at the examples in front of us. We have a radish here on the left, the Tientsin green heritage variety we grew last year, a broad bean variety, and perhaps the hardest of all, a biennial Brassica that we would grow on site for seed. In terms of radish, they are of the Brassica family, so applying what we've learnt about this vegetable type, we know that it is an outbreeder, it's highly variable, and for us at Heritage Seed Library, our selection process involves actually removing them from the ground at edible stage in order to select down, to rogue out poor examples and keep only the very best examples to grow on for seed. In terms of population size for the final harvest stage, ideally the higher the better. Very minimum would be 16 plants, but to be honest, we would aim to be more towards the 100 plants end if possible, and clearly that obviously reflects the starting point in terms of the space you need and the seed that you're going to sow. My recommendation is to isolate and introduce pollinators, which we do using White's fishing maggots. You can hand pollinate. The flowers themselves are self-incompatible, so you do need to transfer pollen across flowers and across plants, and that can be quite time consuming. So in terms of seed production, they're quite easy to grow as an annual, but you do need to factor in the pollination requirements for that. In terms of broad bean, well, just like runner beans, they are prolific cross-pollinators, and I know we looked at some of the isolation requirements and distances for broad beans. They are both partial inbreeders as well, so they do have perfect flowers and they will self-pollinate, but because they will readily cross-pollinate, and one of the things you need to bear in mind, of course, is if you're growing these in a rural area where there may be agricultural crops nearby, they will easily cross-pollinate with field bean. In terms of population size, again, you can get away with 20 to 30 plants. Again, the higher the better, ideally, in terms of seed production. And you do need to look closely at any differences for selection, which may be more difficult with something like a broad bean and a black and white flower generally, but you can certainly make a decision based on the information you know about that particular variety. And finally, Brassicas, well, we looked at the growing requirements for Brassicas. Again, a very high population number, and ideally we're looking at to save seed from around 100 plants by the end of the growing season in the second year. In terms of pollination requirements, as with the radish, the flowers are self-incompatible, and we also introduce pollinators, and we need to grow in a very large space. So we're talking about fruit cage spaces, isolation cages with mesh covers of around 4 meters by 10 meters for our crop requirements. And it's worth noting that there are some open pollinated vegetables that will show greater variation across a particular variety. That could be, for example, land races or selected populations, and it's going to be this background information that's really essential when selecting the next generation for seed production. And I know that for some of you growers, exploring this further, particularly in terms of crops that work well in Wales, is something that you may well be interested in producing seed for. So land races, they will have some similar shared traits, but they may well have a much wider genetic base than some standard varieties. They're very often connected to a particular region where may have been grown for a very long time. They could have freely cross-pollinated between plants within the locality, but generally they'll have been isolated from others by distance. So for example, on the right-hand side there, the Shetland cabbage or kale, that's been grown on the Shetland Islands from around the 17th century and isolated from other cabbage varieties by the sea, but may well have freely cross-pollinated across Shetland. There are marked differences in leaf colour and head formation, but it's still recognised as being very distinct from other cabbages. And of course there are localised versions of the Shetland cabbage, lots of different varieties that have been selected and demonstrate that variation across the region. Selections or populations of vegetable varieties have been bred to have a greater mixture of trait. Sometimes that's often colour. So for example, the sweet corn rainbow sweet Inca, one of the heritage varieties in our collection, has a rainbow range of kernel colours, and that still reflects the fact that it is a population rather than a distinct and stable variety itself. But it's still, in terms of seed production, it needs to be isolated from other varieties outside of that population. Similarly, Bubble and Squeak, that's a squash in our collection that produces green and yellow squashes in that particular variety type. So as seed growers wanting to produce a quality seed crop, you will always be trying to evaluate and account for any variability that you see throughout the growing process, either as evidence of the genetic variability of that open-pollinated variety, or as evidence that in fact cross-pollination has taken place. Remember, going back to our two basic principles. So with highly variable varieties, this isn't an exact science. Knowing that the variety that you're growing is going to be very important, and being able to make a judgment about thresholds in those that variability as either an inbreeder or an outbreeder will also come with a certain level of experience and knowledge of the crops that you're growing. So how do you spot whether we're talking here about evidence of cross-pollination or evidence of something that's off-type, atypical, and needs to be rogued out? Well, we do really have to rely on visual clues and background information to indicate whether cross-pollination has occurred. Certainly for us at the Heritage Seed Library, we're not using anything other than visible information. We do not do genetic testing as a rule on our crops, and we're very much using the methods that have been applied for many, many years in order to do this. So signs that we can look out for could be that there's a significant proportion of vegetables that don't look like the variety characterization information. There could be presence of characteristics that are very different from that information. So it could be anything from different coloured pods or flowers or fruits or their shape. That may indicate that there's been a cross-pollination, especially if there are several different characteristics appearing. The background information is also very important, and before we make a judgment ourselves in terms of what's happening with this variety, we always look back very carefully at the growing history for previous generations of seed, in case they may not have been isolated to the extent that we need them to be. So I've got an example here on the left hand side of one of our heritage varieties. This is Jescot's Longan runner bean. We had some question marks about its seed production, and we decided to grow out a sample of 10 plants. We would normally be growing 30 plants in order to produce the seed, but this is an example of one of the 10 or 20 observational trials that we do each year, and background information suggested that maybe the donor hadn't isolated the crop, although it wasn't very clear. And about 5 to 10% of the plants produced flowers that were bicoloured red and white instead of red. But everything else about that population of plants was showed very little variation, fairly consistent. Pod shapes, pod lengths as described. Similarly, the seeds were of the coat pattern and size, etc., that we were expecting. So from that trial grow out, we were quite happy to accept the fact that the small amount of variation in that population of plants was probably more likely to be down to genetic variability than evidence of cross-pollination more generally. So we were able to proceed with that particular variety as one in our collection. Of course, there are other factors to bear in mind that may demonstrate a difference in appearance, but might not be evidence of cross-pollination or an off-type either. And it could be, it's useful to note that some characteristics, especially flower and pod colour, may change as the plants themselves mature. So we've got some examples here. The Mr. Grease brown flowered broad bean in the centre there has flower colours that change from purple to brown with age, and that difference obviously when you check the characterisation for that variety will depend on the maturity of the flowers at the time that you check that. Many French beans and peas have flowers that will have an initial colour change in the first few weeks, particularly as they mature to producing the pod level. So on the right hand side I've got an example of Mange 2 been growing in my garden, and the flower colour at the top is purple, but by the time it matures to drop its petals, it's changed to a very dark blue navy colour. Similarly, we've got some changes in colour on some of our runner beans, so they may show a very small evidence of coloration. You can see that in the middle there, but that red coloration may change with time as the pods themselves mature. So for the final part of this webinar, we're going to look a little more closely at biennials and look at what the selection and roguing process means for them. So we know from previous webinars that in contrast to annuals, where the plants will flower and set seed in a single growing year, biennials are going to flower and set seed in their second year. They have a much longer exposure time, therefore, to potential pests and diseases. So, for example, there may well be pests that overwinter on the plants themselves. They become hosts for pests to emerge the following spring and summer. So often there's a more planning and cultivation requirements needed for biennial seed production. For us, it's a particular issue this year for our brassicas that we're growing. We've got, for example, cabbage aphid. They're found on the flowers and the green pods in the second year, and we're dealing with them organically with certified biocontrols, in particular lacewing larvae, in order to manage this. Further examples, parsnip moth, for example, is common on wild parsnip and hogweed, and it will feed on the flowers and seed. It's not seen on the parsnips that are grown to eat, but it can be problematic in the second year for vegetables grown for seed. So, as we also discussed in the previous webinar, biennials will overwinter either in the ground or in some form of storage, so there may well be losses during that period of time, and if they fail to re-establish when replanted. So you can see on the right hand side here we've got some beetroot. We may well have selected a population of good roots that were typical of that variety. We will have overwintered them. And we may have lost a few during that process, so perhaps they damaged, or we've discovered some of them have rotted. And when we've planted them out, you can see those little mesh cages around each of the roots. That's to protect from rodents who are particularly interested in these roots at this time of year when they're planted out. But you can also see on the right hand side some of those plants are also not looking too good, so we're likely to lose a few that are not too happy about being replanted back out again. All of those population sizes need to be accommodated. Those potential losses need to be accommodated from the very start of this process, from the number of seeds sown all the way through to the final harvest. That's particularly the case with biennials, where you have more opportunity for losses, as well as them being outbreeders that require roguing out. So we could be looking for something like a cabbage, as we've mentioned before, sowing several hundred seeds before we get to the process of producing a large enough population for the second year seed production. In reality, for biennials, most of your roguing and selection is going to take place after that first year's growth. before you overwinter and before you move into the second year for seed saving. So we've got an example here, one of our heritage varieties, Afghan purple carrot, and the process of selection is illustrated here. So in terms of growing, well, we know a little bit about the variety. We know it's a purple carrot with a very distinctive bright yellow core. We know that on average the carrots are going to be between 20-25 centimetres, allowing for a certain amount of variability in that. And we also have some anecdotal evidence that it may well be resistant to carrot root flies. So in terms of our selection process, well, there's the harvest on the left hand side. Clearly we've grown a very large population in order to be able to rogue down to around 30 to 50 roots for overwintering, and we can happily divide those up based on things that we want to discard as a poorer example of this particular variety. There are other factors that we need to consider, and for us, I think in 2017 we tried growing carrots in their first year. In fact we lost the entire sowing for that particular year because of exceptionally hot weather. Carrots in particular are sensitive to temperatures. They don't really like germinating over 22 degrees, something like that. And while we were growing later in the year, in the summer, we had exceptional heat waves, which certainly made our growing plans much more difficult, and we lost that initial harvest of seed. So we also need to rogue out any diseased plants or anything that's looking damaged, roots that might not overwinter very well, and also rogue out any undesirable traits that we might find in some of these particular carrots. So you can see along the bottom there we've got a whole range of wonky looking vegetables, perfectly healthy, but certainly from a selection point of view we want to be of course saving seed only from the very best examples. So here's a little activity to get us all thinking about what you're going to be looking for when you are selecting your crop, in this case carrots. You're going to be digging them up when they are at the usual edible stage, and you're going to be making some decisions about whether or not you keep these or you rogue them out. So let's work across the examples from the left hand side. So the first one is a white carrot. Obviously for Afghan purple, that's not really what you're looking for at all, and you're going to be roguing them out. You'll also be looking at the proportion at which they appear, because of course you're accounting for genetic variability, but you're also at this point going to be making a decision about whether you are seeing evidence of cross-pollination. So for a white Belgium variety, that may well be the perfect example. Or of course, and something we haven't really talked about in any detail, is that you may well be selecting and developing your own variety. So you may be looking to select for particular desirable features that you then want to develop further. Moving on to the right hand side, forked roots, very common. You've just seen a fine selection of wobbly roots that we would discard. It's a healthy plant, it's a healthy root, but if it is an exception to the rule and other carrots are growing quite happily without forked roots, which are an environmental response, and growing in the same conditions, then why keep it if you can go with the very best examples? Of course, if you are actually stuck for population size, you could overwinter this as an example, but obviously it's not going to be the best example. The further two to the right, the root shape, assuming it's a pointed carrot rather than a blunt shape or a round shape or whatever variety you're looking for, the difference between the two really is just the density of foliage. So you're going to be making a decision, either is it typical of this variety, or in fact is it a desirable feature or less desirable feature for this variety. So dense foliage on the particular carrot might be desirable because it suppresses weeds, or it could be less desirable because it harbours potential pests. So depending on the reason for selection, whether you're developing your own variety or whether it's typical of the variety you're saving seed from, you'll make a decision based on that. Down to the bottom left, again pretty common, a split root. Again, an environmental response. It could well be as a result of too much water or too little water at a particular growing phase. Again, if there are other carrots that aren't demonstrating this feature, then you may want to select those that have a better response to environmental stresses than this particular carrot type. And of course, cracked split carrots, if you're going to overwinter them, there is a higher risk of having potential disease issues with carrots in this state. To the right, hairy carrots. Well, we know certain commercial varieties for consumption, it tends to be less desirable to have a carrot that it requires significant preparation before you can eat it. Hairy carrots could be a particular feature of the variety and it's worth it for the taste, or you might be wanting to select out this particular feature as being less desirable. To the right hand side, clearly you are going to be roguing out plants that don't look healthy. This is a illustration of a unhealthy carrot. You want to be roguing those out, but you also need to be making a judgment about what the issue is with that particular carrot, or even your population crop as in general, but particularly if there are potential risks around seed-borne diseases. And finally, to the right, again, another feature you may see on carrots, green shoulders based on sunlight exposure. It could be fairly typical for that variety, or it could be an undesirable trait that you want to rogue out as an exception. And of course, what you want to end up with is selecting only the very best examples, and ideally you want to have 20 to 30 good roots that have overwintered well, that you're able to plant out the following spring, so that you can ensure that even with losses in the second year, you're going to be saving seed from a minimum of 16 carrot roots. Now, one of the things I haven't talked about really is a notion of genetic drift, and essentially what we are trying to do with open pollinated varieties is be aware that the plants are going to be genetically different each time, which of course allows them to adapt. But from our perspective, we are selecting each time against usually visible features, and there will be some element of genetic drift as that occurs. Now, from our perspective at the Heritage Seed Library, one of the things that we do, and this is a recent example of what we have grown over the last year or so, is we will often grow two different generations of seed, and as old a generation of seed as we possibly can, plus a more recent generation. One, to ensure that we have got a decent genetic gene pool to work with, but also to try and ensure that we are preventing too great a deal of genetic drift if we possibly can. So this is Dobby's purple beetroot. And what's interesting is that this taught us a little bit about our own selection process. So we were able to grow from some quite old seed, in fact 1994, which is pretty remarkable really for beetroot seed. But that population compared to a more recent batch of seed that we grew out showed a much greater purple colouring in the foliage than the majority of the beetroot from the younger seed. Although the root shapes were pretty similar and were what we were expecting to see, what it taught us is that perhaps we had selected out the correct foliage colour in later grow outs. So that's something to bear in mind. We focus particularly on the features that are typical for this variety as a consumable vegetable, but actually it showed that there were some distinct differences likely to be the case of selection rather than any evidence of cross-pollination that we in fact had done ourselves in the process of saving for seed. And finally, selection and roguing or rejection of seed continues all the way through right to the harvest stage, and this very often is the first opportunity for you to see the seed through its complete life cycle, and it's a time to check and further rogue out and select at this point as well. You want to be checking for overall health and the condition of the seed. You will be removing any damaged or undersized seed and just conducting checks for viable seed. So for example, you may be able to use a winnower to select out the best ripe seed. Generally heavier is good, and winnowing will remove any lighter seed that's likely to be less viable or undersized. flattened squash or cucumber seed, for example, could be broken open and you'll find empty seed coats. You want to be checking for pest damage, and for us at the Heritage Seed Library, we're particularly vigilant with broad beans. We want to prevent bean weevil issues by freezing the broad bean seed for at least a week, so that's something you might want to consider. And of course, we also need to be continuing to make this judgment about whether what we're seeing in front of us is evidence of cross-pollination or simple evidence of some genetic variability within that open pollinated variety. So a couple of examples here are recapping really on what we picked up on the second webinar. Is what is it that you're seeing in front of you and what judgment are you going to make on it? Firstly, to the left, it's a French bean variety and it's displaying what we typically tolerate as inversion of seed coat colour. So that is not evidence of cross-pollination as such, but just some genetic variability, and in fact may well be a feature of that particular variety. To the right, however, is a black-seeded runner bean, and while we can tolerate differences as an open pollinated variety, generally we're looking at thresholds of between 5 and 10% variability, whether that's flower colour, pod shape, and in this case the seed coat, final seed coat colour. For us, we would be pretty suspicious that this is evidence of cross-pollination in a previous generation, which is rather unfortunate if you've spent the year growing this. And again, I talked about pre-checking seed in the second webinar to make sure that you aren't seeing evidence of cross-pollination in the previous generation before you have already sown out. So an important stage, the harvest stage for seed production, and it is as important to continue the selection process at this point. So over the course of these 3 webinars, we've looked at seed production and the fact that maintaining a variety so that it is true to type in future generations needs 2 simple rules. We are going to try and prevent cross-pollination and remove those undesirable individuals along the way. And while we looked at cultivation requirements specifically in webinars 1 and 2, this process of selection that we've covered will help ensure that you produce a good quality crop of seeds. All the information about population sizes, whether a vegetable type is an outbreeder or an inbreeder, and the cultivation requirements you need to use to grow for seed are available on the Garden Organic website. You need to follow the Growing Advice tab through to the Seed Saving Guidelines, where you'll get vegetable specific information. And of course, the Tyfu Cymru website itself has this webinar as well as other resources to help support your commercial enterprise. So thank you so much for listening to me, Diolch yn fawr, and good luck with your seed growing plans.
That was brilliant. Thank you very much, Katrina. That was a great presentation. We do have a couple of minutes now just for questions, if you want to stay around for those. We have got a couple of questions here already, but if there's any on your mind, please type them in the Q&A box and we can get them across to Katrina. So the first question, when is the best time to lift roots like carrots and beets to overwinter to save?
Okay, we always lift them at the end of the growing season when they are at what we would consider to be the edible stage. So typically we sow carrot and beetroots a little later in the year, probably sort of beginning of June time for us. Obviously it depends on your growing season, and then we will be lifting them later in the year when we would normally lift them to grow them. That reason is obviously we do some characterization checks then, but that's the time when we would typically lift the roots.
Brilliant. A question here about roguing. How important is timing when roguing out? And are some decisions more time critical than others? Yeah, so we've kind of covered the idea that there is a difference in maturity and that will affect its appearance against what you're expecting to see. But there are cases where you do need to
look at them at a very specific time. So for inbreeders which are at low risk of cross-pollination, you can afford to get to the pod stage or the fruit stage before you notice any difference. For some biennial crops, as we've just said, your most important selection period is going to be when you're picking them up and digging them up at harvest stage, when you would normally eat them. But for example, if you have a cross-pollinating outbreeder that might be insect pollinated, the flowering stage is particularly important. So if you spot on a bush squash that one of your plants is actually a trailing squash, but the flowers have already appeared, you have to assume that some cross-pollination may have already occurred. So and similarly with runner beans, if the flower types are all wrong on one particular plant, but the flowers are now exposed and they will have been visited by bees, if you haven't isolated them, then you do have to assume that cross-pollination then will occur at that point. And you don't want that process of pollination to happen and produce the same feature in future seed. You can reset the clock, and that happens quite a lot at the Heritage Seed Library. If we find that we've got an exposed stigma on a tomato variety, for example, then we will simply remove the flowers, remove the plant if you're talking about roguing out, and reset the clock so that they can reflower and start again.
Brilliant. And this question's from me, actually. I was really interested in what you said about freezing seeds. Is that something you do as standard at the Heritage Seed Library? Is it just for, you mentioned with beans, is that something you do with other varieties? And is there an optimum temperature, and is there any risk with freezing? Is there something you have to consider if you're going to do that?
Yeah, actually all very good questions, and we're experimenting a little bit at the moment. So there are two reasons why we freeze seed at the Heritage Seed Library. One is, as I've mentioned, with the broad beans in particular, kill off anything that might be inside the seed that we don't want it to emerge, like the bean weevil, for example. You can only do that if the seed is extremely dry. So we only do it with a broad bean. At a particular stage. Very often what we do is we dry it further before we freeze it. So if we harvest it, if we're not sure, or we always take this precaution ourselves, we often isolate that seed harvest from any other seed, often under a mesh cover itself, and dry it out further before we put it in frozen, because seed that's not perfectly dry, it doesn't like it. Obviously the water crystals will damage the seed itself. In terms of freezing generally, well, we have a black box system, so that's a backup system for our collection, to make sure that if we all went up in flames, we've got some starting point for any of our seed in our collection. So we have a black box collection at Warwick University, which has seed samples from the majority of our varieties. We keep continuously adding to it, and the freezing temperature is -18 to -20 degrees. Again, we further dry that out for at least another couple of weeks, just to make sure it's definitely below 5% moisture level, and then that's good then to freeze, depending on the vegetable type, 30 years or longer for some seed. So that's our kind of insurance policy. And we're also experimenting to see if we can extend the shelf life of some of our seed harvests. So I keep talking about our Brassicas, and obviously they're quite tricky for us, but they also produce an enormous amount of seed, usually for us way beyond the distribution demand from our members. And obviously when you've gone to so much trouble to produce that amount of seed, we've been experimenting to see whether freezing will help further extend that shelf life. For some things, for Brassicas, that's pretty good. For other things, it's less so, and we're doing particular experiments on certain French bean varieties at the moment, because we found that's quite variable.
Yeah, brilliant. Very, very interesting stuff. I think that's all the questions that we've got from everybody. So thank you very much again for your presentation today, and for the seed programme that you've helped us deliver. We do have some more webinars coming up that I just wanted to let everybody know about. If you're in the ornamental side, we have a plant protection ornamentals coming up on the 18th of August. We are running these breakfast bite-sized webinars, which are focused with improving your online visibility. So we've got looking at Google Analytics on the 21st of August and how to make minor changes to your website to get found on Google, and that's on the 4th of September. And then we also have a peat-free horticulture how and why webinar, which is happening on the 28th of September. So that seems a way off yet, but please register on the website if that is something you're interested in learning more about. And obviously, if you've got any questions about this webinar or anything else coming up or anything Tyfu Cymru can support you with, then please get in touch with us. Just want to say, when you finish this webinar, a form will come up. We would love to hear your feedback. So if you could complete that, that would be fantastic. And that's it from me. Thank you very much for joining us today.